If you have ever taken a DNA test and discovered Mexican ancestry, what does it really mean? Have you wondered what are the roots of this genetic finding? Are Mestizos and indigenous Mexicans now two genetically different groups, or do they share the same ancient heritage? What scientists have discovered about Mexican genetic origins is so extraordinary that it shatters every assumption about ancestry in the Americas. The answers hidden in Mexican DNA reveal a story more complex and mysterious than anyone imagined. Deep in the rainforests of Yucatan, Quintana Roo, and Chiapas, the Maya peoples have preserved one of the most distinctive genetic signatures in all of Mexico. Modern Maya populations, including the Tzotzil, Tzeltal, Chuj, Canjobal, and the isolated Lacandan, carry mitochondrial DNA patterns that tell a story of extraordinary demographic continuity stretching back thousands of years. Maya populations display a unique genetic component that reaches 80 to 90% frequency in southeastern Mexico, found primarily in Mayan-speaking groups. This Mayan component appears at 10 to 20% levels in central Mexican natives, suggesting ancient gene flow connections between regions that had persisted for millennia despite geographical barriers. The mitochondrial DNA profile of Maya groups shows distinctive patterns within the four major Native American haplogroups: haplogroup A2, B2, C1, and D1. But what makes Maya genetics truly mysterious is the presence of specific sublineages within these haplogroups that appear nowhere else in Mexico. Maya Y chromosome patterns reveal equally intriguing complexity. The paternal lineages are dominated by haplogroup QM3, but Maya populations harbour specific Q sublineages, including QZ768 and QL663, that show divergence times suggesting separation from other Native American populations, thousands of years before conventional archaeological timelines would predict. The Lacandon Maya present the most extreme case of genetic isolation. Living in the Lacandon rainforest, near the Guatemalan border, this small population of fewer than 1,000 individuals shows genetic patterns unlike any other group in Mexico. Their mitochondrial DNA displays founder effects so severe that nearly 60% of individuals belong to a single maternal lineage. Yet this lineage carries mutations that suggest isolation for over 2,000 years. Most remarkably, Maya populations from different regions show genetic connections that span political boundaries, suggesting that Maya genetic identity transcends modern nation-states and reflects ancient population movements that occurred long before European contact. The genetic legacy of the Aztec Empire presents one of Mexico's most complex demographic puzzles. The Nahua peoples, direct descendants of the Aztec ruling class, are distributed across multiple states including Mexico, Puebla, Veracruz, and Hidalgo. Yet each Nahua population displays distinct genetic characteristics that reflect both ancient diversity and more recent population movements. Nahua populations studied from different regions show remarkable genetic variation that challenges simple assumptions about ethnic unity. The average number of singletons per individual was higher in Nahua populations, reflecting their descent from the largest civilizations in Mesoamerica. Central Mexican Nahua groups display mitochondrial DNA compositions, with haplogroup A2 reaching 35 to 40%, higher than in northern populations. However, the Nahua Coyolilo population from Veracruz presents a striking anomaly. This group shows 42% European ancestry and 22% African ancestry, despite speaking Nahuatl and maintaining indigenous cultural practices. Y chromosome analysis reveals the complexity of Aztec descendant populations. Native Mexican populations from the Valley of Mexico, including Nahuas, Otomis, Popolocas, Tepehuas, and Totonacas, show enormous paternal genetic diversity. This diversity suggests that the Aztec Empire incorporated populations from across Mesoamerica, creating a genetic melting pot that persists today. The Otomi people, who coexisted with the Aztecs throughout central Mexico, present their own genetic mysteries. Otomi populations from Hidalgo show mitochondrial patterns that cluster with other central Mexican groups, yet their Y chromosome signatures include unique Q sublineages that suggest they maintained distinct paternal lineages despite centuries of contact with neighboring populations. Ancient DNA from Teotihuacan, the great pre-Aztec metropolis, has revealed that urban centers in central Mexico maintained extraordinary genetic diversity. Individual burial sites contained people with mitochondrial lineages spanning the entire range of Native American variation, suggesting that cities like Teotihuacan attracted populations from across the Americas, while somehow preserving their genetic distinctness. The indigenous populations of northern Mexico, the Tarahumara, Huichol, Pima, Seri, and others, inhabit some of the most challenging environments in North America, and their genetic profiles reflect both ancient isolation and remarkable demographic resilience. The Tarahumara, also known as Raramuri, occupy the rugged Sierra Madre Occidental of Chihuahua and have developed one of the most distinctive genetic signatures in Mexico. Y chromosome analysis reveals that Tarahumara males carry predominantly QM3 lineages, but with specific sublineages that appear to have evolved in isolation for thousands of years. Tarahumara mitochondrial DNA shows a unique pattern, with haplogroup C1 reaching 35 to 40%, significantly higher than in most other Mexican populations. Haplogroups A, 2, and B, 2 each appear at 20 to 25%, while D1 reaches 15 to 20%. Remarkably, some Tarahumara individuals carry the rare haplogroup X6, which appears elsewhere only in the Huichol population, suggesting ancient connections between these northern groups. The Huichol people of Jalisco and Nayarit present one of the most genetically isolated populations in Mexico. Studies of Y-linked short tandem repeats revealed that Huichols show the lowest gene diversity among Mexican indigenous groups, demonstrating the effects of genetic drift attributable to geographic isolation and founder effects. The presence of 2 principal paternal lineages supports historical records indicating that Huichols were formed by the fusion of 2 ancestral Mexican tribes. Huichol mitochondrial DNA displays similar founder effects, with haplogroups B and C being more common than in central Mexican populations. The presence of haplogroup X6 in 3 out of 15 Huichol individuals studied represents one of the highest frequencies of this rare lineage found anywhere in the Americas. The Seri people of Sonora represent perhaps the most genetically isolated indigenous population in Mexico. Living along the Gulf of California coast, the Seri have maintained genetic patterns that suggest extreme isolation for over 1,000 years. Their long-term effective population size was calculated at only 984 individuals, the smallest of any Mexican indigenous group studied. Seri populations show the highest proportion of runs of homozygosity among all Mexican indigenous groups, indicating that their high genetic differentiation results from genetic drift rather than admixture. Despite their coastal location, Seri genetics show little influence from other Native American populations, suggesting they maintained cultural and biological isolation even from neighboring indigenous groups. The Pima populations of northwestern Mexico present a contrasting pattern. Y chromosome studies showed that Pima and Tarahumara indicated clear differentiation from other indigenous Mesoamerican populations such as Mixtec, Otomi, Nahuas, and Purepecha, supporting the division between Arido-American and Mesoamerican populations that occurred roughly 4,000 to 9,900 years ago. The ancient civilizations of Oaxaca have left genetic signatures that rival the Maya in their complexity and antiquity. The Zapotec and Mixtec peoples, creators of Monte Alban and other monumental centers, harbored genetic diversity that suggests their ancestors were among the earliest inhabitants of Mesoamerica. Zapotec populations show mitochondrial DNA patterns that form a cluster with indigenous populations living in an area from central Mesoamerica to Central America. Complete mitogenome analysis revealed that Zapotec and Mazawa populations shared no sequences in common, while each showed a number of unique subclades, indicating long periods of independent evolution. One Mazahua individual carried the D4H3, a subclade of haplogroup D4, a lineage that has been mostly found in South America along the Pacific coast. This finding suggests ancient migration corridors that connected Mexico with South America through pathways that remain largely unknown to archaeologists. Zapotec Y chromosome patterns show the same complexity observed in their mitochondrial DNA. Populations from different Zapotec communities in Oaxaca display distinct paternal lineages within the broader QM3 haplogroup, including sublineages that appear to be specific to the Zapotec region. The Mixtec people, neighbors, and sometimes rivals of the Zapotec, show genetic patterns that reflect both their shared Oaxacan heritage and their distinct cultural development. Mixtec populations from different regions, Mixteca Alta and Mixteca Baja, display genetic differences that correspond to geographical and cultural divisions that have persisted for over 1,000 years. Modern genetic studies have identified a multi-ethnic component that is prevalent among Otomanguean linguistic family populations, including both Zapotec and Mixtec groups. This component reaches its highest frequencies in the center and south of Mexico, suggesting ancient population movements that connected Oaxacan peoples with groups across Mesoamerica. Along Mexico's Pacific coast, from Sinaloa to Chiapas, indigenous populations display genetic patterns that suggest this region served as a crucial corridor for ancient population movements between North and South America. The Mayo, Huichol coastal settlements and various smaller groups harbor genetic signatures that bridge the gap between northern desert peoples and southern Mesoamerican civilizations. The Mayo people of Sinaloa and Sonora show genetic affinities that distinguish them from both pure Arido-American and Mesoamerican populations. Phylogenetic analysis based on mitochondrial DNA control regions shows that Mayo populations cluster with Mazahua and Pima groups, separate from the cluster including Zapotec, Maya, Tepehua, Nahua, Huichol, and Otomis in Mesoamerica. Coastal Huichol settlements show genetic patterns distinct from their mountain relatives, suggesting that environmental adaptation and geographical isolation created population substructure, even within single ethnic groups. These coastal populations show evidence of ancient connections with South American populations that can't be explained by recent contact. Y chromosome analysis reveals that Pacific coast populations harbor specific Q sublineages that appear to represent ancient migration routes along the coast. Some of these lineages show closer relationships to South American populations than to other Mexican groups, supporting theories of coastal migration routes during the initial peopling of the Americas. The Gulf of Mexico coast presents a completely different genetic landscape, shaped by the interaction of indigenous populations, with successive waves of migration from both the Caribbean and the African diaspora. The Totonac, Huastec, and other groups of Veracruz show genetic complexity that reflects Mexico's position as a crossroads of global migration. Totonac populations display mitochondrial DNA patterns that include all four major Native American haplogroups, but with frequencies that differ significantly from other Mexican indigenous groups. Totonaco populations from Veracruz show genetic patterns that align more closely with South American populations than with some other Mexican groups when tested with ancient DNA samples. The Huastec people of northern Veracruz and southern Tamaulipas present one of the most linguistically isolated groups in Mexico. They speak a Mayan language despite being separated from other Maya populations by hundreds of kilometers. Their genetic patterns reflect this isolation, showing affinities with other Mayan groups despite their geographical separation. In addition to the standard QM3 lineages found throughout indigenous Mexico, some populations show evidence of pre-Columbian contact with Caribbean populations, including Y chromosome lineages that may have arrived through ancient maritime connections. The presence of haplogroup L in some Gulf Coast populations, particularly in Nahuacoyolilo, provides direct evidence of African genetic contributions that arrived through colonial-era demographic processes. This population shows 22% African ancestry, despite maintaining indigenous language and cultural practices. The mountainous regions of central Mexico harbor indigenous populations that have maintained genetic isolation despite their proximity to major urban centers. The Purepecha of Michoacan, the Otomi of Hidalgo, and other highland groups show genetic patterns that reflect both ancient population structure and more recent demographic events. The Purepecha present one of the most genetically diverse indigenous populations in Mexico, with wide distribution of haplotypes along constructed network joining trees, suggesting that their present genetic composition was influenced by Purepecha dominance in western Mesoamerica during pre-Columbian times. Purepecha mitochondrial DNA shows a balanced representation of all four major Native American haplogroups, with no single lineage showing the founder effects observed in more isolated populations. The Tepehua population, whose language belongs to the Totonac family, shows genetic affinities with the Huastec due to their shared history, yet displays differentiation from Otomi groups and Maya-speaking Chols. Huichol and Tarahumara populations, both belonging to the Uto-Aztecan language family, show closer genetic relationships than either shows to other Uto-Aztecan speakers, supporting their linguistic classification. However, some Nahua populations, also Uto-Aztecan speakers, show genetic patterns more similar to Otomanguean speakers. Recent ancient DNA studies have revealed that many Mexican indigenous populations carry genetic signatures from ghost populations, ancestral groups that contributed significantly to modern genetics, but left no archaeological or historical trace. These ghost lineages appear within specific indigenous groups and provide clues about Mexico's hidden demographic history. Ancient DNA analysis identified two distinct, unsampled genetic ancestries that contributed to pre-Hispanic populations of northern and central Mexico. One group appears to be related to modern mixed populations from southern Mexico, but the other represents something entirely new, a genetic lineage that suggests human presence in Mexico far longer than conventional chronologies allow. Some indigenous populations show evidence of multiple ghost population contributions. The discovery of ghost populations within indigenous groups challenges fundamental assumptions about population continuity in Mexico. If entire ancestral populations could disappear while leaving only genetic traces in modern groups, it suggests that Mexico's demographic history is far more complex than archaeological evidence indicates. The 1921 census, conducted after the Mexican Revolution, asserted that 59.3% of Mexicans self-identified as Mestizo, 29.1% as indigenous, and only 9.8% as white. These figures became essential to cementing the mestizaje ideology that shaped Mexican identity through the 20th century. Yet modern genetic analysis revealed these classifications bore little relationship to biological reality. Recent scrutiny by historians and activists has questioned whether such drastic demographic changes from the 1793 census were possible, citing the relatively low frequency of interracial marriages in colonial Mexico. The discrepancies were staggering. Some populations classified as Mestizo showed genetic frequencies similar to continental Europeans, Durango, or European-derived Americans, Jalisco, while others classified as indigenous, based on language, showed higher European admixture than some Mestizo populations. Various authors theorize that the Mestizo identity promoted by the government led to people who are not biologically Mestizos being classified as such, creating a fundamental disconnect between cultural identity and genetic reality. Beyond genetic mysteries lay equally puzzling patterns in visible traits. A University College London study found that 18% of Mexicans had blonde hair and 28% had light eyes, frequencies that seem to contradict simple admixture expectations. American Sociological Association research revealed that differences in blonde hair frequency between northern Mexico, 22.3 to 23.9%, and central regions, 18.9 to 21.3%, were less pronounced than commonly believed. Studies of blood type frequencies provided another window into genetic complexity. Research has found that foreign blood groups, A, B, AB, were more common in northern and western Mexico, while the O blood type, universal in indigenous Americans, dominated southern regions. Since indigenous populations exclusively exhibit O blood type, the presence of other groups indicated foreign genetic influence. Most remarkably, 51.8% of Mexicans showed Mongolian spots. The genetic composition of Mexican Mestizos represents one of the most complex admixture patterns in human population genetics, reflecting 5 centuries of demographic transformation that created a new type of human ancestry, unlike anything seen elsewhere in the world. Recent comprehensive genetic studies have revealed that Mestizos are not simply a straightforward mixture of 3 ancestral groups, but rather harbor genetic complexity that varies dramatically across regions, social classes, and even individual families. According to the most recent large-scale autosomal DNA studies, Mexican Mestizos show an average genetic composition of 55.2% Native American, 41.8% European, 1.8% African, and 1.2% Asian ancestry. However, these averages mask extraordinary regional variation that reflects the complex history of settlement and admixture across Mexico. A study with 300 self-identified Mestizos revealed that Sonora showed the highest European admixture at 61.6%, while Guerrero displayed the lowest European component at only 28.5%. The geographical patterns of Mestizo ancestry follow predictable gradients that reflect historical settlement patterns. Genetic estimators revealed that Native American ancestry ranges from 37.8% in northern Mexico to 81.5% in southeastern regions, while European ancestry ranges from 11.5% in the southeast to 62.6% in northern Mexico. But perhaps the most striking aspect of Mestizo genetics is the dramatic difference between maternal and paternal ancestry patterns. While autosomal DNA shows relatively balanced Native American and European contributions, Y chromosome studies reveal a completely different story. The paternal ancestry of Mexican Mestizos is predominantly European at 64.9%, followed by Native American at 30.8% and African at 4.2%. This pattern becomes even more extreme in northern and western regions, where European Y chromosome frequencies reach 66.7% to 95%. In stark contrast, mitochondrial DNA studies show that almost 90% of Mexican maternal lineages belong to indigenous American haplogroups A2, B2, C1, and D1. This extraordinary sex-biased admixture pattern suggests that Mexican mestizo populations formed primarily through unions between European men and indigenous women, creating a demographic transformation that was both systematic and sustained across centuries. Socioeconomic stratification adds another layer of complexity to mestizo genetics. Studies correlating genetic ancestry with social class have revealed systematic patterns that suggest genetic sorting mechanisms operating across centuries. Low-income mestizos in Mexico City showed 59% Native American, 34.8% European, and 16.2% African ancestry, while higher socioeconomic mestizos averaged around 70% European ancestry. This correlation between genetic ancestry and social status reflects historical processes that continue to influence contemporary Mexican society. Medical genetic studies have revealed that mestizo genetic diversity has profound health implications. The dramatic difference in lung capacity between mestizos with Western versus Eastern indigenous ancestry is so pronounced that equally healthy individuals of the same age can appear a decade apart in lung function tests. The Jewish presence in Mexico represents a hidden chapter of the country's genetic history, involving multiple waves of migration that have left distinct genetic signatures in both clearly identified Jewish communities and the broader Mexican population. Current estimates place Mexico's Jewish population at 80,000 to 90,000 individuals, about 75% concentrated in Mexico City. But genetic studies suggest that Jewish ancestry extends far beyond self-identified Jewish communities. Mexico's genetic legacy stands as testament to the extraordinary resilience, adaptability, and diversity of our species, preserving in living DNA the stories of migrations, adaptations, encounters, and transformations that shaped not just one nation but the entire human experience in the New World.